In this video, we'll discuss about immunofluorescence. What is fluorescent? It is a type of dye. It is a special properties. It absorbs the light of wavelength. It absorbs light of UV and emit rays of visible light. So fluorescent absorb UV light and emit visible light. Fluorescent is a technique in which it will take antibody. We will labeled with fluorescent dye. We will labeled with fluorescent dye. Wherever it will go, dye will go it. It will emit light. This is antigen. This is antigen. Here putting antibody over it. Antibody labeled with the fluorescent dye. It will emit light. It will be visible in the form of fluorescent light. So it will emit light. So we will able to detect the presence of antigen or antibody. That is the basic fundamental idea. There are two types of fluorescent dyes. One is fluorescein isothiocyanate. It is blue green color, and rhodamine. It is orange red fluorescent color. So two type of dyes examples: fluorescein isothiocyanate, it is blue green. Then rhodamine, it is orange red color. Types of immunofluorescent: there are two types: direct fluorescent and indirect fluorescent. Direct fluorescence used for detection of antigen in specimen, mainly detection of rabies virus. Then indirect fluorescent used for detection of antibody in specimen, mainly used for detection of syphilis. So direct fluorescent method. In this method, take tissue on slide. Suppose particular antigen present in that what we are suspecting. It can be present or not. Suppose present means what we will put fluorescent labeled antibody to that particular antigen. Example, rabies in the brain sample. Here antigen rabies. We will take anti-rabies antibody labeled with fluorescence. We will put it over the slide. It will remain for 10 minutes. After that we will wash it. If antigen present, bond with antigen. If not present, it is washed away. So after washing, see under microscope whether antigen present or not. If light is emitted, means antibody present on the tissue only, means antigen present on slide. If light is absent, the antibody is washed away because no antigen is present. So that specimen suspecting positive labeled with antibody, fluorescence observed, antigen is presented in the specimen. Light presented means antigen present. Light absent means that antigen is absent. Light presented means antigen present. Light absent means antigen absent. So it is a direct method. Fluorescent just emitting light. Now coming to uses. What are the uses? Most important, it is sensitive method to diagnosis rabies by detection of rabies virus antigen in brain smear, commonly employed for detection of bacteria, virus, other antigens in blood, CSF, urine, feces, tissue and other specimen. The only disadvantage, separate specific fluorescent labeled antibody. Whatever antigen you are suspecting, to take corresponding antibody. If you suspect rabies, take anti-rabies antibody. If you suspect herpes, take anti-herpes. If you suspect HIV, take anti-HIV antibody. So we will take corresponding antibody and label it. Labeling is very costly. Labeling is very costly. Coming to second type, the indirect fluorescent technique employed for detection of antibody in serum or other body fluid. Here we will take a slide. In this one we will apply patient blood samples, that syphilis patient blood samples. If antibody are presented means bind with antigen. Third things antibody against antibody. We will label it. So in this phenomenon, take a slide, take suspected antigen, then primary antibody. It is presented means antigen antibody binding will occur. After that put secondary antibody. It is labeled. Again we will wash it. After that we will observe under microscope. So after washing, see under microscope. If primary antibody absent means secondary antibody will go in washing. If color presented means positive, absent means negative. So what are the uses? Detection of antibody in serum of syphilis patient. Rabies used for direct method, syphilis we are used for indirect method. What is the advantage? Here only antibody against antibody. So again and again labeling is not required. So indirect is the best method. This one is the statement format. Thanks for watching.